When we consider the safety of the human brain, we often imagine a physical trauma. The importance of wearing helmets, the dangers of concussions, the role of the skull as a built-in shield. But what about threats that attack from within the brain? Each of the conditions listed on this slide have some sort of biological mechanism. They target the brain at the level of individual brain cells by disrupting their activity or their access to resources and launch their attack from the inside out. Unlike a physical injury, these conditions can't be protected against using a helmet or a skull, which begs the question, how does the brain defend itself? It may surprise you to know the protagonist of today's story is in fact a net, specifically a perineuronal net, or PNN, depicted here in green. Now, as the name suggests, these nets surround individual neurons, forming a protective barrier. Whereas most nets are made up of threads and fibers, perineuronal nets are composed of proteins and sugars, but they share the same basic structure. Molecules are layered and intertwined, swaddling the neuron and giving the net its uniquely web-like structure. And it's this incredibly intricate structure which allows the net to regulate two essential properties: plasticity and stability. Plasticity is an expression referring to how malleable our brains are, how easily our neurons can change shape or form new connections. Essentially, plasticity determines how easy it is for us to learn. Children, for example, have especially plastic brains, making it easy for them to learn new skills or new languages. Adult brains, on the other hand, are a little bit less plastic, making it harder for us to absorb new information. Now, a good way to understand the role of PNNs in plasticity is by thinking of the brain as a ball of clay. Now, this clay will take on different forms throughout different stages of life, but during early childhood, when the brain is the most plastic, we can think of it as a ball of wet clay, flexible and ready to be molded. Now, as we get older and we acquire new knowledge, our brains will begin to take on a new shape. The more mature our brain is, the more defined that shape will be, until eventually it takes on what will more or less be its final form. This bowl of clay, then, is analogous to a fully mature brain. Now, the role of the PNN in this process is to regulate plasticity. That is, to determine how plastic the brain is at each stage of life. You see, as the PNN sets in, and its many layers of webbing wrap around the neuron, it actually makes it increasingly difficult for the cell to change shape or form new connections. In other words, with an increase in PNNs comes a decrease in plasticity. In the context of our clay brain example, this means that the more PNNs we have, the drier or less malleable our clay becomes. Now, you might be wondering, why on earth would anyone want a less plastic brain? What's the point of these nets if all they do is make it more difficult for us to learn? And the answer to this question lies in the second PNN-regulated function, namely stability. You see, although plasticity is great when it comes to learning new things, much like malleability is ideal when sculpting clay, it also makes it much more difficult or much easier to forget what we've learned. When plastic, the brain is destabilized. Neurons are free to form and reform as many connections as they wish, so nothing's ever actually preserved. This is not the case once PNN set in. In surrounding the neuron, they restrict its growth, yes, but they also protect it from degradation, from neurotoxins, and they provide it with much needed stability. As you can see then, these two properties are at odds, each one compromising the other. Too much plasticity and brain activity becomes dysregulated, leaving us vulnerable to conditions like schizophrenia. Too much stability, on the other hand, and the brain becomes rigid, making it difficult for us to learn or change our pattern of thought, as seen in depression. It's the responsibility of the perineuronal net to strike a balance between the two, making adjustments as needed throughout every stage of life. In studying these nets, then, we gain insight as to how the brain changes with time and how we might develop therapies for conditions such as Alzheimer's. With all of this in mind, then, I'd like to circle back to our original question. How does the brain protect itself? And I hope by now you know the answer is simple: by relying on its built-in safety nets. Thank you.
So how do you study the net? Do you do imaging, or do you do molecular biology, or what are you using in terms of techniques?
Yeah, many students in my lab use microscopic imaging. So we study, for example, the density of these nets in different brain regions in different conditions. So by imaging the nets using microscopes, we can say there are more nets in one specific area in, for example, Alzheimer's disease, or fewer nets in another area in a different type of condition. In my specific project, actually, I do molecular biology. So what I look at is the genetic expression of the neurons beneath the nets themselves. So I look to see whether there are different genes being expressed in these specific neurons in individuals who have suffered very severe child abuse. So that's the specific area that I look into.
Rappel, hein, c'est ici et maintenant que vous votez pour Dominique pour le prix du public. Une autre question dans la salle? Oui?
Allô? Tu as parlé des liens entre les réseaux périneuronaux et aussi, dans le fond, certaines maladies comme la dépression. Mais est-ce qu'il y a certains traitements qui existent qu'on voit que ça passe par ce réseau-là aussi pour répondre à la maladie? Par exemple, est-ce qu'il y a des antidépresseurs qui agissent sur le réseau périneuronal pour faire leur action?
It's actually a really interesting question because, c'est correct si je réponds en anglais?
Yes.
Yeah. Very interesting question because one of the starting studies that allowed us to understand that there is a link between these nets and depression was one in which they observed that when you apply antidepressants, when you prescribe antidepressants, you actually see a reduction in perineuronal nets. So the medication is not the intention, but it seems to be a product which led us to believe or led us to suspect that an increase in perineuronal nets could contribute to the pathogenesis of depression itself.
Oui?
How can you observe if the nets are more like for stability or plasticity?
How do we observe that?
Yeah, like how do you know which one it has more if it's like dysfunctional?
It depends on the maturity of the perineuronal nets. So when the nets are more immature, they're more prone to encourage plasticity. So when they first start out, they're not fully formed. There's a lot of holes in them. It's a very literal web-like structure. They're kind of responsible for like a controlled plasticity. So the neuron isn't able to change shape completely, but they allow a little bit of flexibility. But once the neuron itself matures, the nets become much denser. So they kind of restrict the cell, quite literally again. And that's why they're encouraging more stability. They're holding it up. The connections that it's formed during its maturation, they sustain those throughout essentially the entire lifespan, if possible. And if not, then that's where you start to see illnesses come into play. I hope that answers your question. Okay.
Question dans la salle? Moi, j'en ai une pour vous, Dominique. Est-ce qu'il y a une façon, vous parliez de stabilité et de plasticité, et c'est un ou l'autre, est-ce qu'il y a une façon d'inciter notre cerveau à aller plus dans une direction que l'autre?
Ben, essentiellement, never mind,
Okay,
sorry.
Yeah.
The more PNNs we have, the more stability we have. So as we get older, we develop more and more PNNs. We tend to go more in the direction of stability with age. But there are enzymes, so proteins, that we can apply to brain tissue that actually eats out these nets. Like I think someone mentioned before, these enzymes are like Pac-Man. So if we apply these enzymes, you eat up the nets, there's a lot less stability, but there's more plasticity. So we've seen in animal studies, for example, if we apply these enzymes, we eat up the nets, they have more plasticity, they're better able to learn new things. So we can see the effect of removing these nets, it actually increases stability. Having more nets increases plasticity, whereas having more nets increases stability.
And is this process being used therapeutically, or it's just experimental for now?
In the context of animals, it's more experimental. We've been trying to get it more into regular therapy, but the difficult part is you don't want to wipe out all of the nets, so you have to know exactly how to remove enough that you protect yourself, but not so many that you're vulnerable to other conditions.